Hello students. Welcome to Dr. Satish Polshettiwar EduDose, your trusted channel for pharmacy and life science learning. Today, I'm going to walk you through a fascinating and foundational topic in microbiology: staining of bacteria, including its types, step-by-step procedures, and how we bring these tiny organisms to life under the microscope. Let's dive into the colorful world of microbes. So what exactly is bacterial staining? Bacteria are incredibly tiny and almost transparent under a regular microscope. To actually see them, we use special dyes to stain and highlight their size, shape, arrangement, and other features. Basically, staining makes the invisible visible. Now let's break down the types of staining, starting with simple staining. This one's pretty straightforward. You use just one dye, like methylene blue or crystal violet. Why? To observe the basic shape and arrangement of bacteria. For example, you can easily spot rod-shaped E. coli or spherical Staphylococcus. All bacteria show up the same color, so it's quick and easy, but you don't get much detail. Next up is differential staining. This uses more than one dye, and the most famous technique here is gram staining. Other differential stains include acid-fast capsule and endospore staining. The goal? To tell bacteria apart based on their cell walls or special structures. So how do you prep your bacteria for staining? First, grab a clean glass slide. With a sterile loop, transfer a tiny amount of bacterial culture, either from a broth or a colony. Spread it into a thin smear on the slide, then let it air dry. Heat fix it gently over a flame. This step kills the bacteria and sticks them to the slide. Just don't overheat or you'll mess up the cells. Now onto gram staining, invented by Hans Christian Gram back in 1884. This classic technique separates bacteria into two big groups. Gram-positive bacteria have thick peptidoglycan walls, hold onto the crystal violet stain, and look purple. Like Staphylococcus aureus, gram-negative bacteria have thinner walls and an extra membrane. They lose the crystal violet and pick up safranin, so they look pink or red. Think E. coli. Here's the gram staining process in a nutshell. First, crystal violet, all cells turn purple. Next, iodine forms a complex with the dye. Then, alcohol or acetone. This decolorizes gram-negatives but not gram-positives. Finally, safranin. Gram-negatives now show up pink. Just like that, one stain splits bacteria into two categories. Once you're done staining, it's time to check out your handiwork under the microscope. Start with low magnification, then switch to the 100x oil immersion lens for sharp detail. Add a drop of oil to the slide. You'll see purple cocci in clusters for gram-positives and pink rods or chains for gram-negatives. It's honestly pretty satisfying. There are also some special staining techniques worth knowing. Capsule stains, like India ink, show a clear halo around bacteria such as Klebsiella pneumoniae. Endospore stains use malachite green and heat to highlight tough spores with vegetative cells showing up pink, think Bacillus species. And acid-fast stains like Zeal-Nielsen pick out Mycobacterium tuberculosis. Acid-fast cells turn red, while non-acid-fast ones go blue or green. So staining isn't just about making bacteria colorful. It's about unlocking their identity, understanding their structure, and revealing their secrets. Here's what to remember. Simple stain, one dye, basic info. Differential, compare and contrast. Gram stain, the gold standard in clinical labs. Special stains, a deeper dive into microbial mysteries. If you learned something new, hit like, share, and subscribe to Dr. Satish Polshettiwar EduDose for more microbiology essentials. Got questions or want a deep dive on acid-fast or endospore staining? Drop a comment below. Until next time, stay curious, stay scientific.
If you found this video helpful, please subscribe, like, and share it with others who'd benefit.